Imagine a disease that has, haunted your family for generations. Just imagine it being rewritten out of your blood in a single afternoon.
I mean, it sounds like pure science fiction, right?
It really does. But welcome to Science After Dark. Tonight, we're doing a deep dive into a quiet, almost invisible revolution that is happening right inside our DNA.
Here we're talking about CRISPR,
Hmm.
and we want to explore it not as some, you know, sterile laboratory concept, but as the actual active power to rewrite the code of life itself.
Exactly, because
Yeah.
I always look at human DNA as this incredibly vast, ancient manuscript, and for decades, the absolute best we could do was just passively read it.
Right, we could sequence it, we could look at it.
Yeah, we mapped the genome, we could spell out all the genetic letters, and we could see exactly where the typos were, the ones causing devastating diseases. But the text was basically under glass.
We absolutely couldn't touch it.
Right, and suddenly we haven't just, smashed the glass. We've actually developed a highly precise search and replace function for the book of life.
And that transition from passive reading to active editing is really the crux of the whole revolution. Our mission tonight is to trace how a bizarre, almost accidental quirk of bacterial biology was reverse engineered by humans to make that happen.
Because it didn't start with human medicine at all, did it?
No, not even close.
Yeah.
To understand the mechanics of this search and replace function, we really have to look at the organisms that drafted the original blueprints billions of years ago.
Yeah.
The entire system comes from bacteria.
Wow. So it wasn't some grand ambition to cure human disease in a lab.
Not at all.
Yeah.
It actually started in 1987 as a complete anomaly in a data set. Yoshizumi Ishino's team in Japan, they were sequencing a completely unrelated gene in E. coli.
Oh, right, E. coli. And they just stumble onto a stretch of DNA that made absolutely no structural sense, didn't they?
Yeah, it was completely baffling to them.
Yeah.
They saw this sequence of short, identical, repetitive DNA palindromes. And between each of these identical repeats, there was the unique sequence, which we now call a spacer.
A spacer. So it would be like reading a book where every other paragraph is exactly the same, but the sentences wedged between those identical paragraphs are just chaotic and totally unique.
That is a perfect way to describe it. And at the time, I mean, they just published the anomaly, essentially noted how strange it was, and moved on. The technology simply didn't exist to decipher it back then.
So it just sat there. It sat completely dormant in the scientific literature for years.
Basically, yeah. Until Francisco Mojica picked up the thread in 1993. And what's really fascinating is he wasn't looking at E. coli at all.
Right, he was looking at something completely different.
Exactly. He was studying Halosirax mediterraneae, which is this microbe that thrives in extreme salt environments. And he found those exact same weird repeating patterns.
Wait, in a completely different organism.
Yes. And his obsession with these structures over the next decade is really what propelled the whole field forward. He and Ruud Janssen eventually coined the name for it in 2001 or 2002. They called it CRISPR.
Which stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a mouthful.
It's a huge mouthful, yeah. But the acronym perfectly describes the physical architecture of the DNA. Even so, the actual biological function remained a total void until around 2005.
That was the big breakthrough year, right?
It was massive. That year is when 3 independent teams, led by Mojica, Christine Courcelle, and Alexander Bulletin, they cracked the code of those random spacer sequences that were sandwiched between the repeats.
Right. They realized those spacers weren't just random genetic junk. They actually matched the DNA of bacteriophages.
Exactly. Bacteriophages being the viruses that actively hunt and kill bacteria. So they realized the bacteria were actively taking snippets of DNA from the viruses that attack them and integrating those snippets directly into their own genome.
It is wild. It's like a localized, highly specific molecular memory.
It really is. And by 2007, researchers at a food company, they were working with the bacteria used to culture yogurt. Actually, they proved this experimentally.
Yogurt bacteria, you really can't make this stuff up.
Right. But they showed that if a bacterium survives a viral attack, it captures a piece of the viral DNA, inserts it into its CRISPR array, and uses it to gain immunity against that specific virus forever.
Okay, so wait, they essentially evolved a biological wanted poster system. Like, the bacteria survive an attack, grab a mugshot of the virus, and literally pin it up in their own DNA.
Yes, a literal mugshot.
And if that exact virus ever comes back, the bacteria's internal security system recognizes the mugshot instantly and just shreds the invader.
Exactly. It is a programmable, highly adaptive immune system. And the very moment that mechanism was fully understood, the entire trajectory of modern genetics shifted.
Because everyone realized the implications.
Oh, instantly. The immediate question became, well, if a bacterial cell can program the system to find and cut specific viral DNA, could human scientists extract the system, reprogram the wanted poster, and force it to cut any DNA sequence we choose?
And that leap, going from a bacterial defense mechanism to a programmable human engineering tool, happened blindingly fast. I mean, by 2008 and 2011, you had people like Luciano Marafini and Emmanuel Charpentier figuring out the crucial RNA components.
Yeah, the groundwork was being laid rapidly. And then came 2012, the Landbar paper from Jennifer Doudna and Emmanuel Charpentier. And Virginie Giussani's independently made the same breakthrough around the exact same time.
They actually pulled the system out of the cell and simplified it into those programmable molecular scissors we always hear about.
Right. And we should really break down the mechanics of what they engineered, because the true elegance of CRISPR is in its simplicity. The system they designed relies on just two core components.
I lay it out for us.
First, you have the Cas9 protein. This is a nuclease, which is just an enzyme whose sole biological function is to cleave or cut the double helix of DNA.
So Cas9 is the physical scissors.
Exactly. And second, you have the guide RNA. This is a synthetic molecule. It's only about 20 nucleotides long, and scientists can literally design it on a computer and order it for a few dollars.
So the guide RNA is the address.
Yes. You attach this guide RNA to the Cas9 protein, and when you introduce this little complex into an environment with DNA, the guide RNA actively scans the entire genome until it finds the exact complementary sequence.
And once it finds it.
Once the guide RNA binds to its target, it essentially pulls the trigger on the Cas9 protein, causing it to make a clean double-strand cut right there.
But wait, there's a vital safety mechanism built into this naturally, right? Because if the guide RNA is just looking for a match, wouldn't the Cas9 end up cutting the bacteria's own wanted poster?
That is a great question.
Because the bacteria has the viral sequence sitting inside its own genome. It obviously needs a way to avoid shredding itself.
Yeah, the bacteria would just destroy its own DNA. So the fail-safe against that is called the PAM sequence, the protospacer adjacent motif.
The PAM sequence.
Right. For the standard Cas9 enzyme, the PAM is a very strict 3-nucleotide sequence, NGG. That's any nucleotide, followed by 2 guanines.
Okay, NGJ.
The Cas9 protein fundamentally cannot bind and cut the DNA unless it physically detects that exact NGG sequence sitting immediately adjacent to the target site.
Oh, I see.
And the bacteria's own CRISPR array, the place where it stores the mugshots, it doesn't have that PAM sequence next to them. So the scissors stay closed when scanning its own DNA.
So it really is like a biological zip code. The guide RNA can have the exact street address, it can find the right house on the DNA strand, but the molecular scissors physically will not open the envelope unless that 3-letter zip code is stamped right next to it.
That is exactly it. And the requirement of that zip code does dictate where we can make edits. But because NGG appears incredibly frequently all across the human genome, it's actually a very minor constraint for us.
Still, proving this worked in a test tube in 2012 was one thing. But moving it into mammalian cells, that has to be a completely different level of complexity. I mean, human cells have a nucleus, they have intense, complex defense mechanisms.
Oh, absolutely. Taking it from a test tube to a living human cell was a massive hurdle. But January 2013 was the tipping point for that.
That was Feng Zhang and George Church, right?
Yes. They simultaneously published work showing that CRISPR could successfully edit the genomes of living human and mouse cells. Zhang's contribution in particular was vital because he optimized the delivery architecture.
Delivery architecture meaning how you actually get the scissors inside the cell.
Exactly. You have to shuttle the Cas9 and the guide RNA through the cellular membrane and then through the nuclear membrane into the nucleus where the DNA actually resides without the cell destroying it.
But here is where the mechanics get really interesting to me. Because just walking up and cutting a strand of DNA doesn't actually cure a disease, does it? I mean, a cut is just damage. So the actual editing relies entirely on how the cell panics and tries to repair that damage once the cut is made.
You're completely right. You're touching on the cell's endogenous or natural repair pathways. We basically hijack those pathways to make the edits permanent.
So how does that work?
Well, when Cas9 causes that double-strand break, the cell detects catastrophic damage. It usually triggers a process called non-homologous end joining, or NHEJ.
NHEJ, okay.
In this pathway, proteins rush to the break site, they grab the severed ends of the DNA, and they essentially just glue them back together as fast as physically possible.
It's a frantic, messy patch job.
Very messy. He usually ends up deleting or adding a few random genetic letters right at the break site, which completely ruins the reading frame of that gene.
So the gene is knocked out, it ceases to function.
Exactly, it's disabled. And this is incredibly therapeutic if your goal is to silence a gene that is actively causing a disease.
Right.
But if you want to fix a mutation to actually rewrite the text, you have to rely on the second repair pathway.
Which is homology-directed repair, or HDR.
Right. And this is where we provide a template. We don't just send in the scissors, we send in the scissors along with a pristine, corrected piece of DNA.
Okay, so we give the cell an instruction manual.
Exactly. If you flood the area with a synthetic DNA template that matches the sequences surrounding the cut, the cell will often use your template to rebuild the broken segment. It meticulously stitches the sequence back together, incorporating your desired edits perfectly.
Hold on, though. You mentioned earlier that the guide RNA costs like a few dollars to synthesize.
Yeah, very cheap.
If a technology that fundamentally alters the code of life is this cheap and this easy to program, especially compared to older, wildly expensive tools like zinc fingers, doesn't that democratization invite massive security risks? I mean, if virtually any molecular biology lab can afford to run this, who is actually regulating the edits?
Yeah, that friction between accessibility and security is one of the heaviest debates in the entire field. The shockingly low barrier to entry is exactly what allowed researchers globally to adapt the technology almost overnight.
True, it exploded.
Right, but that exact same democratization is what accelerated CRISPR out of just academic theory and directly into clinical application. The timeline from understanding the basic mechanism to actually deploying it in human therapies was compressed into just a decade.
Which is unheard of in medicine. And that timer basically went off in late 2023, early 2024. We saw the first regulatory approvals for a CRISPR-based therapy called Casgevy.
Yes, targeting sickle cell disease and beta thalassemia. The clinical rollout of Casgevy is just a masterclass in exploiting those cellular repair pathways we just discussed.
And it happens ex vivo, right? Outside the patient's body.
Correct. Physicians extract blood stem cells from the patient's bone marrow. Then, in the laboratory, they use Cas9 to make a very precise cut.
But wait, I was looking at this, and they aren't actually using the precise HDR pathway to fix the broken adult hemoglobin gene that causes sickle cell. They're doing something much more indirect.
That's the genius of it. They're using the messy NATJ pathway to just break a switch.
Break a switch.
Yeah, so before you are born, your body produces fetal hemoglobin, which is highly efficient at carrying oxygen. But shortly after birth, a genetic enhancer called BCL11A kicks in.
Okay.
And this enhancer shuts off the fetal hemoglobin factory, forcing your body to transition to adult hemoglobin. Now, in sickle cell patients, that adult hemoglobin is defective.
So instead of trying to perfectly repair the defective adult gene, they just use the CRISPR scissors to chop up the BCL11A enhancer.
Exactly. They break the off switch.
Wow. So the switch is destroyed, and when those edited stem cells are infused back into the patient, they repopulate the bone marrow and just start churning out pristine fetal hemoglobin again.
Yes. The fetal hemoglobin entirely compensates for the defective adult version. It effectively cures a disease that causes just unimaginable lifelong agony.
It's brilliant. It's like bypassing a collapsed highway by just forcing a forgotten dirt road back open.
That's a really good analogy, yeah.
But, we have to address the shadow hanging over this. Because the conditioning required to clear the patient's bone marrow before you put the edited cells back, it's grueling. And the price tag for Casgevy sits at roughly $2.2 million per patient.
It is incredibly expensive.
We're talking about a reality where the technology exists to cure the incurable, but the economics threaten to create a future of profound genetic inequality. Are we moving toward a society where only the ultra-wealthy can afford a genetically clean bill of health?
The economics of personalized genetic medicine are definitely daunting. And the applications are only expanding. I mean, CRISPR is being heavily deployed in oncology right now.
Right, for cancer therapies.
Yeah, like in CAR T-cell therapies. Researchers extract a patient's immune cells and use CRISPR to knock out the PD-1 gene.
What does the PD-1 gene do?
It acts as a brake pedal on the immune system, which tumors actually exploit to evade detection. So by destroying that brake pedal, the immune cells become hyper-aggressive against the cancer. They started doing this in China back in 2016.
And outside of medicine, the agricultural applications are just bypassing traditional regulatory hurdles entirely.
Oh, completely.
We're seeing non-browning mushrooms from 2016, Japanese tomatoes engineered to lower blood pressure, cattle edited to grow slick coats so they survive extreme heat.
And the reason regulators are lenient is because researchers are using CRISPR to just delete or tweak existing letters. They aren't introducing foreign DNA from other species.
Right, so to the regulatory bodies, a deletion made by a Cas9 enzyme in a lab is functionally indistinguishable from a deletion caused by, natural solar radiation in a field.
Exactly. But whenever you have a technology this malleable, the temptation to cross established ethical boundaries becomes overwhelming.
Which brings us to the dark chapter.
Yes, for CRISPR, that boundary was fundamentally shattered in November 2018.
This is when the drive to eliminate disease basically mutated into the temptation to perfect the human genome.
Yeah. At a genome editing summit in Hong Kong, Chinese biophysicist He Jiankui announced he'd used CRISPR to edit human embryos, which were subsequently implanted. The result was the birth of twin girls, known by the pseudonyms Lulu and Nana.
He was targeting the CCR5 gene, wasn't he? Trying to introduce a mutation that makes human cells resistant to HIV infections.
That was his goal, yes. But the global reaction was absolute unified condemnation. He violated fundamental scientific norms and crossed a biological threshold that the international community had explicitly agreed not to touch.
Because there is a massive, massive difference between editing a patient's blood cells and editing an embryo. It's the line between somatic editing and germline editing.
Precisely. When we talk about therapies like Casgevy, we're talking about somatic cells, the blood, the liver, the lungs. If you edit a patient's somatic cells, the changes are entirely confined to that individual.
Right, they're cured, but their reproductive cells remain completely untouched, meaning they cannot pass those edits on to their children. So using our manuscript analogy from earlier, somatic editing is like taking a bottle of whiteout and fixing a typo in one single printed copy of a book. It repairs that one specific volume, but it obviously doesn't impact any other book in existence.
But germline editing alters the sperm, the eggs, or the early embryo itself. Those edits don't just dictate the biology of the resulting child, they become a permanent fixture in the human gene pool.
Because they'll be inherited by that child's offspring.
And their offspring, indefinitely.
So germline editing is like walking into the publisher's warehouse and physically altering the original metal printing plates. Every single copy of the book printed from that day forward will carry your change forever.
It's a permanent alteration to human evolution. And the scientific community found He Jiankui's work not just ethically reckless, but biologically deeply flawed.
Really? How so?
Because the CRISPR machinery was injected after the embryo had already begun to divide, the edits did not penetrate every cell equally. This resulted in something called mosaicism.
Meaning the twins have a patchwork of edited and unedited cells.
Exactly. Furthermore, while disabling that CCR5 receptor might offer HIV resistance, we know that receptor plays a crucial role in fighting off other pathogens, like the West Nile virus.
Wow. It perfectly illustrates why we can't just hack complex biological systems without understanding the cascading consequences. I know he was sentenced to 3 years in prison, heavily fined, and it triggered multiple global summits pushing for strict governance.
Yes, the WHO and others stepped in. The consensus remains that clinical germline editing is currently far too risky and ethically unresolved.
But the immense clinical value of somatic editing has triggered an entirely different kind of conflict, hasn't it? The battle over intellectual property.
Oh, the patent war. It has been brutal.
Yeah, there has been this fragmented, fierce patent war over the foundational Cas9 technology between UC Berkeley representing Doudna and Charpentier, who actually won the 2020 Nobel, and the Broad Institute, representing Feng Zhang's eukaryotic applications.
And while the institutions fight over the patents for those original scissors, the actual scientists have moved on. They're building entirely new machines.
Because as revolutionary as Cas9 is, it still relies on physically shattering the double helix, which carries the risk of off-target cuts.
Right. Off-target effects were the primary bottleneck for years. Sometimes the guide RNA finds a sequence that is, say, a 95% match, and the Cas9 enzyme mistakenly makes a cut there anyway.
And in a genome of 3 billion letters, accidentally severing the wrong sequence could inadvertently trigger oncogenes and literally cause cancer.
Exactly. So they've developed incredible detection methods like guide-C-Sec.
How does that work?
It specifically tags any double-strand break in the cell with a short, synthetic piece of DNA. This allows researchers to sequence the genome and map exactly where Cas9 is going rogue so they can re-engineer safer guide RNAs.
That's clever.
But honestly, the most profound leap has come from researchers realizing that cutting both strands of the DNA might not be necessary at all.
Right. This is where the whole molecular scissors analogy officially breaks down.
It really does. In 2016, David Liu's lab at Broad Institute unveiled base editing.
Base editing.
Yeah. They engineered a version of Cas9 that is disabled. It's a nickase. It only nicks one side of the DNA ladder instead of breaking it in half. And they fused this disabled Cas9 to a completely different enzyme, a deaminase.
So what does that hybrid machine actually do?
It walks up to the target sequence and chemically converts a single nucleotide base, say a cytosine, directly into a uracil. The cell's own replication machinery then naturally reads that uracil and replaces it with a thymine.
That is staggering. It doesn't break the ladder. It literally just reaches in and chemically transmutes one letter into another.
Exactly. And then in 2019, they pushed it even further with trim editing. They fused a Cas9 nickase to an enzyme called reverse transcriptase.
And what does prime editing do?
It acts as a true, literal search and replace function. It reads a customized RNA template and writes entirely new genetic information directly into the DNA strand. It safely bypasses those chaotic cellular panic pathways entirely.
It's basically a genetic word processor equipped with a highly accurate molecular spell checker.
That's exactly what it is. And from the trial data, this isn't theoretical anymore. Companies like Intellia and Verve are moving these editors into in vivo clinical trials.
Meaning they don't even extract the cells first.
Right. They're using lipid nanoparticles, which are just microscopic fat bubbles that protect the delicate CRISPR machinery in the bloodstream, to deliver these advanced base editors directly into the livers of living patients to correct severe genetic mutations.
It is breathtaking to think we've gone from puzzling over bizarre repetitive patterns in E. coli DNA to literally chemically transmuting the code of life inside a living patient's liver.
The sheer speed of this field brings us to a tension that is absolutely going to define our century.
Yeah.
Because on one hand, we possess this beautiful, unparalleled promise to eradicate genetic suffering that has plagued families since the dawn of humanity. But on the other hand, we hold the terrifying responsibility of directing our own evolution. Somatic editing is a triumph of modern medicine. But the germline frontier, the power to re-engineer the human gene pool for all future generations, is a threshold we are still standing right in front of.
Which leaves us with a final thought for you to turn over in the quiet hours tonight. If humanity eventually masters these molecular word processors enough to completely eradicate all hereditary diseases, do we lose a fundamental part of human biological diversity? Do we owe it to future generations to give them a genetically clean slate, or is the line between healing a patient and engineering a human too blurry to ever cross safely?
It is the defining question of our time.
Thank you so much for exploring these depths with us tonight. Keep questioning, keep wondering, and have a wonderful night. We are your hosts, and this has been Science After Dark.